Hi, we are going to continue to talk about viral multiplication. The first phase we defined as adsorption, which is attachment. The second phase was penetration and uncoating, so now it's in. So now we have viral nucleic acid inside the cell. What happens now? The next phase is synthesis. So let's talk about what we mean by synthesis. All right, synthesis is going to be making the needed proteins that are going to be needed to make more virions and also copying the nucleic acid for that. So the viral nucleic acid is inside. Now, we do need to understand that there's some differences about how different viruses go about doing that. If the virus contains DNA, so remember, viruses can have RNA, could have DNA. If it is a DNA virus and the viral nucleic acid is DNA, that viral nucleic acid will move into that viral DNA, will move into the nucleus. It stays in the nucleus, it is replicated there, and it is assembled there. All right, DNA viruses moving into the nucleus. That makes sense because that's where all the host DNA is. That's where all the enzymes that are needed to replicate host DNA are, and we know our virus is going to use the host machinery, the host enzymes. All right, so DNA viruses are moving in the nucleus. The RNA viruses actually stay in the cytoplasm, which also makes sense because the majority of the host RNA work in the cytoplasm. So the RNA viruses are going to get replicated there and assembled there. Now, there is a category of viruses called retroviruses. These are RNA viruses, but instead of staying in the cytoplasm and behaving like other RNA viruses, they actually move into the DNA, into the nucleus, and then they use those enzymes and an enzyme they bring with them called reverse transcriptase to copy themselves from RNA into DNA. And then they behave more like DNA viruses. So that's an example for that. All right, so remember, if a virus is going to need an enzyme right at the very beginning, it needs to get packaged with that, like reverse transcriptase. Otherwise, it gets there, uses host enzymes, or synthesizes its enzymes using the host ribosomes and the host machinery. Okay, so synthesis. So what happens? We've got DNA viruses, we've got RNA viruses, and they behave differently. So let's talk about DNA viruses first. Where are they? They went to the nucleus, right? Because that's where all the DNA is, the host DNA, and that's where all the enzymes needed to copy themselves are. So they're in the nucleus. Double-stranded DNA and single-stranded DNA behave a little differently. At the very beginning, moves into the nucleus, and then if it's double-stranded DNA, it behaves pretty much the same way as host cell DNA gets copied and transcribed into mRNA. The host cell has double-stranded DNA. It knows what to do with it. So if the virus is double-stranded DNA, pretty much it's the same as the host. If it is single-stranded DNA, now this is something our host cell doesn't have to deal with very much. It actually has to kind of fold upon itself a little bit, and it creates a primer, and then it can, and it makes itself into double-stranded DNA, and then it behaves using the host cell replication machinery. All right, so that all happens in the early phase. The very beginning, it will start making mRNA of enzymes that it needs in the early stages to replicate itself and to make those proteins it might need at the beginning. They do define late phase after it gets some of these proteins made, then it will start making proteins needed maybe to form the capsid, those capsomeres. It'll start transcribing those proteins. All right, so that's double-stranded DNA viruses and single-stranded DNA viruses. What about those RNA viruses? RNA viruses are going to stay where? Cytoplasm, because they're RNA, so they're going to stay in the cytoplasm. They'll either be single-stranded or double-stranded RNA. If it's single-stranded, we actually classify them as either positive single-stranded RNA or negative single-stranded RNA. What's the difference? A positive single-stranded RNA virus is equivalent to mRNA, messenger RNA. And you know that that's what gets taken to the ribosomes and translated and makes a functional protein. So if the viral nucleic acid is positive single-stranded RNA, it's uncoated, and now I have this positive strand, it can attach straight to a ribosome and start being translated and start making viral proteins. So some viruses come in as positive single-stranded RNA, and they can be directly translated into proteins. All right. If it comes in as a negative single-stranded RNA, then it's going to have to make a copy of itself. It's going to have to use complementary base pairing to make a positive single-stranded RNA before it can make a protein. Either way, we also, in addition to making proteins, we also need to replicate this strand of RNA. Whatever was packaged in the virion is what needs to be packaged to go out. So a positive single-stranded RNA needs to be replicated and make a whole bunch more positive single-stranded RNA. Same thing here. This negative single-stranded RNA needs to make a whole bunch of negative single-stranded. The only way to do that is that template process. So let me show you here on my little whiteboard that I drew out. If it is positive single-stranded RNA, this is just a strand of nucleic acid with bases, guanine, cytosine, adenine, uracil, because it's a RNA here. It is going to use complementary base pairing, and the machinery is going to come in and lay down a match. And you're going to get a C and a G and a U and an A, and it's going to lay down that match. So we can make a whole bunch of this. From this copied, and this is referred to as a negative single-stranded RNA. If it is a positive single-stranded RNA and we need to make a whole bunch of this to get packaged in virions, it makes this template, and then this template uses the same complementary base pairing to make a whole bunch of this to go in. So that's how that is working. Okay, so either way, we need to make positive single-stranded RNA to be used as messenger RNA on both sides, and we need to make templates to create the nucleic acid that gets packaged. Now, double-stranded RNA, it actually, what we have observed is double-stranded RNA gets unzipped and pulled apart, and then once it's separated, it serves as positive single-stranded RNA and negative single-stranded RNA. It gets copied, and then those pieces come together. All right, so that is synthesis, and then the next video will be on assembly. Thanks.